In this video we will discuss about this integrin activation through cell signaling. So basically how this integrin ligand binding domain gets activated through cell signaling. So what are the different kind of cell signalings that cause this integrin ligand binding domain activations is shown on the left hand side. It can be either chemokine receptors, CD3 T cell receptors or toll-like receptors and other selectins. So what happens when this receptors binds to its chemokine or its respective ligand, and it will trigger intercellular signalings, and those intercellular signalings will cause this transformation of this Ref1 GDP to the Ref1 GTP. So basically addition of one phosphate, it will cause a Ref1 GTP, and this Ref1 GTP either alone or in combination with RIAM will change the conformation of this talin from closed talins to the elongated talin, and then these elongated talins can bind to the beta domain of this integrin. Integrin without intercellular signaling activations is generally is in band closed form, and once this talin interacts with this beta subunit of the integrins, it will change the conformation from band close to extended close. However, the ligand binding domain is still not active. So in order to activate that ligand binding domain, this kindlin needs to bind to the beta unit of this beta subunit of the integrins, and once this kindlin and talin binds, then it will change the conformation from extended close to extended open, and this is quite stable conditions where these integrins can now bind to the its counter receptors. So this is one of the reasons, this is one of the way by which these integrins can be activated, and the changes that occurs in from the extended close to extended open is that this talin will also help in the separation of the alpha this alpha subunit of the integrins and the beta subunit of the integrins. As we can see that the distance between alpha and beta subunits increased in the extended open conditions compared to the extended close conditions. So this once the integrins are activated, it can then also helps the tumor cells in the tumor evasions or tumor metastasis and migrations, and those things we have seen in one of the paper where we had discussed about the selectin interactions with the integrin subunits. Select on the immune cells, interactions with the integrin subunits on the tumor cells, thereby preventing the immune cells to attack the tumor cells. So those things are also can be correlated with this integrin activations. That integrin activations increased in the case of tumor cells, and thereby they can interact with the immuno checkpoint or the glycoimmuno checkpoints on the immune cells, and they triggers the immune evasion or the immune suppressions. So, and the chemokines can, one of the example of this chemokine receptors could be this TGF beta, as TGF beta is known to cause this mesochemical to epithelial characteristics, or not mesochemical, epithelial to mesochemical characteristics through this MAD pathway. So in case where all the genes that are involved in the TGF beta initiated EMT, the all the genes mediated proteins needs to be present. All the genes that are involved in the SMAD pathway needs to be present so that it can interact or it can trigger these epigenetic changes, and that will cause the epithelial to mesochemical transition. However, in the absence of this any of the genes that are involved in this downstream signaling, it will not trigger EMT through their respective chemokines. For example, in the case of KPN1, since SMAD4 is absent, even though in the presence of TGF-beta, will not trigger the epigenetic changes or the epithelial to mesochemical transition. We might have to use different cytokines that does not use the SMAD pathway for EMT. So I hope you understood how this integrin activation works and what are some of its applications in terms of the tumor cells and the immune cells interactions. If you need to understand more about this topic in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.